Hello everyone, I'm sitting here with Dr. Steve Horvath, who is a professor of genetics and biostatistics at UCLA. Probably one of Steve's most well-known contributions to biology is the development of what's known as the Horvath Aging Clock. And since then, he's gone on to develop even more accurate aging clocks, which I'm so excited to talk about. I've talked about your work multiple times on multiple podcasts to multiple scientists. So thank you for spending time to talk with me today.
Thank you for your interest. Thank you for visiting me.
Well, Steve, maybe we could kind of start at the beginning with what is this Horvath aging clock that you had developed? Like, can you explain what it is?
Well, the Horvath aging clock is what I sometimes call the so-called pan-tissue epigenetic clock. And so it is the most accurate molecular measure of age. It applies to all cells in the bodies, certainly all cells that have DNA, all tissues, all organs. It measures age in prenatal samples, in children, all the way to supercentenarians, people who are over 110 years old. So it measures age. So if somebody provides, for example, DNA from blood or DNA from neurons or DNA extracted from saliva or urine, I can very accurately estimate their age.
Their chronological age, like how old they are in years.
Exactly, and that's already a deep question. So the clock does measure chronological age. However, it's of course not a perfect measure of chronological age. There's always an error. For example, if I analyze the blood from a 50-year-old, the epigenetic clock may say, well, this person is actually 55 or 45. And so there's a small error. And this error is actually biologically meaningful. You know, it's not just noise, but rather it is in part related to what people call biological age.
That's super interesting. There, that basically the error was actually related to biological aging, because that was the next thing I was going to say was, you know, people age at different rates. Like even, you know, obviously chronologically they could be the same age.
Yes.
But if you look at a variety of biomarkers, in fact, there was a paper published a few years ago in PNAS that looked at like 18 different biomarkers.
Yes.
They looked at glycated hemoglobin, so HbA1c, VO2 max, triglycerides, telomere length, immunosenescence, a lot of the, you know, typical biomarkers that are clinically used to like determine health status.
Yes.
And it was basically found that people, you know, aged at very different rates based on those biomarkers. So some people biologically appeared much younger than their chronological age, and some people appeared much older than their chronological age.
Exactly.
So how now you've developed a different clock that can actually predict?
That's right. Well, let me comment first on the term biologic age. It's a very intuitive term. Most people have a vague understanding of it. It somewhat relates to morbidity risk or mortality risk and also aging. But strictly speaking, it's not well defined because different researchers have different ways of measuring biological age. Some people use clinical markers that you mentioned, various markers of glucose levels or lipid levels and so on. Now, my take to measuring biologic aging is based on a chemical modification of the DNA molecule. It's DNA methylation, you know. So, and I mention it because depending on how you measure biologic age, you might get different answers. So a person might look bad in terms of glucose levels, and you would say, well, they age faster than they should. However, it could turn out that according to methylation, they are actually in pretty good shape, you know. So, yeah.
Have you seen that before, where you can see people have, for example, like other clinical biomarkers that are not that are unhealthy, like higher fasting blood glucose, or maybe elevated triglycerides, elevated C-reactive protein, a marker of inflammation?
Yes.
Do you find that those typically correlate well with the epigenetic age, or?
I wouldn't say it correlates well. It correlates, you know, so people who have higher levels of inflammation and what you mentioned, their epigenetic clock goes a little bit faster, but the word is there's a weak relationship, you know, because it is quite possible that somebody turns out to be in good shape according to epigenetic aging rates. And the number one example I want to mention in this context are actually people of Hispanic ancestry. Unfortunately, Hispanics often have a higher risk for diabetes, the higher metabolic syndrome. And however, according to the epigenetic clock, they actually age more slowly, you know. And so there's this disconnect. And this is actually an interesting disconnect because there's something known as the Hispanic mortality paradox, you know. Hispanics, as I mentioned, have often a disadvantageous risk profile according to clinical biomarkers, but it turns out on average they live much longer than expected. They actually live longer lives than people of European ancestry, you know. And that association is paradoxical to a clinician who looks at clinical biomarkers. But according to the epigenetic clock, it's not paradoxical because, as I mentioned, we have found that Hispanics age more slowly according to the epigenetic clock.
That's very interesting. So in this case, the epigenetic clock correlates more closely with lifespan than with clinical biomarkers of health status.
That's right, yeah.
Well, in a way, that's kind of good because, I mean...
Well, it shows it adds something, you know. So the epigenetic clock is actually very much under genetic control. So some people just inherit a genome that makes, or DNA that really allows their epigenetic clock to progress more slowly. And so the heritability is about 40%, you know. And yeah, so in this sense, it's not just lifestyle factors. By contrast, some of the clinical biomarkers you mentioned are very much under the influence of lifestyle, you know. So you can probably cure high glucose levels by just avoiding carbs, right? And and also high lipid levels by avoiding or taking statins, you know. So there are a lot of clinical markers that can be influenced with lifestyle interventions and pills. By contrast, we don't have many interventions that allow us to reverse the epigenetic aging rate. You know.
But do we know that? We haven't been testing that though, right?
Yeah, I mean, not really. I mean, in my lab, we clearly want to find interventions that slow the epigenetic clock. And by now, many people are working on it, you know, and it's a gold rush. Who comes up with an intervention that affects the epigenetic clock, you know? But yeah.
So you mentioned that the heritability was about 40% of that. Is that of the epigenetic aging clock,
yes.
or okay? Yes. So you, because I recall from your looking at the epigenetic clock of these semi-supercentenarians, people that are like 105 years old.
Yes.
They had epigenetic aging clocks that were like 8.6 years younger than their actual chronological
that's true.
age.
That's a manifestation of that. So if you have a parent who lived until age 100 or 105, then chances are that your blood is actually younger than the blood of a person of the same age, same gender, same everything, but whose parents didn't live until age 100, you know. So the offspring of centenarians obviously have a genetic advantage, hopefully, but also that is manifested in the epigenetic clock.
Yeah, so you see that their epigenetic aging seems to be slower.
Yes. So that's one line of evidence, but there's another. So people have these longitudinal epidemiological studies, and they may have collected a blood sample from a person when they were, let's say, 40 years old. And then 15 years later, they get a second blood sample, you know. And so you can then ask the question whether a person who was aging quickly at the first blood draw, did they still age quickly at the second blood draw, you know? And the answer is yes. And conversely, you observe the same for people who have who age more slowly, you know. And that, in my opinion, this could already be observed in when you study, let's say, a person at a young age 20. You draw their blood, then if you followed them for 60 years, you know, you would find this consistency that people who are slow agers at age 20, they are also slow agers at age 60 or 80. You know.
So that does seem to imply that genetic.
Yes.
I mean, unless someone dramatically changes their lifestyle. How stable are these changes in methylation? These methylation patterns that are so-called the aging clock.
Yes.
How stable are they over a person's lifetime? I mean,
they are remarkably stable. So when we compare to any other genomic measurement, I mean, they would be far more stable than anything I'm aware of. They're more stable, far more stable than gene expression, proteomics, metabolomics measurements. All the omics are less stable, you know. And that's really the biological reason why these epigenetic clocks are the most accurate measures of aging. It's just that methylation is so stable, you know. And it's stable not just in vivo, but also when people collect DNA, so we've collected DNA and then we didn't store the blood tubes properly, so they melted, you know. And we extracted DNA and the measurements were perfect, you know. So so even on a technical level, they're very stable.
That's interesting, because I've done experiments intentionally, though, almost the same, where we were collecting blood samples from participants in a trial, and I was measuring DNA damage as biomarked by gamma H2AX. And I wanted to know how long I could have blood at room temperature before I started to get artifactual DNA damage happening.
Yes.
And so I did time course, and found that after 2 hours of blood being at room temperature, there's just tons of DNA damage that started to increase.
Yeah,
that
that's
was
scary. That's
right.
Scary.
So
that wouldn't be the case with DNA methylation. I know, because I hired a phlebotomist here in LA to visit families to collect blood, and this person didn't have an air conditioner in his car, and it was the hottest day in LA ever. So all the blood tube melted, you know. That was my experiment. And then I just couldn't send this phlebotomist out to go back to the families to collect blood, you know. I felt sorry for the family, so that's why we did this experiment, you know.
Yeah.
And all I can tell you, we got beautiful data. And so I see that over and over. And people sometimes ask me, what if you have a forensic sample, you know? So let's say a blood stain, you know, that was, let's say, in a room for a couple of weeks, or even let's say a bone sample collected, you know, a couple of months later. All of these data, in my opinion, would still lead to very good methylation measurements for the purpose of measuring aging.
Yeah, because it's so stable.
It's just so stable.
Do you find that because these methylation patterns obviously are changing with age, you were able to predict first chronological age with pretty 96% or so accuracy?
Yes.
And so, obviously while they're stable, at the same time they are changing.
Yes.
But do they change, like, is there a pattern of change? Like, do they change, every few years, all of a sudden things rearrange? Or is there, like, a pattern you can see where things start to change every, like, block of time?
Yeah.
Do you know what I'm saying?
Yeah, I do. It's a good question, So our epigenetic, let me start slowly and say the epigenetic clocks typically track several hundred locations in the genome, For example, the pan-tissue clock is based on 353 locations in the genome. And a question is whether each locus changes, each location gains methylation in a continuous fashion, and probably not, So I think what happens is some locations gain methylation, others lose methylation, and it's a bit random. But on aggregate, once you average hundreds of sites, you kind of average out the noise, the variability, and that's that gives rise then to this very accurate age estimate. You know?
Okay. I definitely want to jump into some questions on mechanism and stuff too, but
Yes.
before I kind of go into the woods, this predicting the biological age, your DNA methylation PhenoAge,
Yes.
was, if I recall from reading your papers, which I read recently, was able to predict all-cause mortality,
Yes.
disease-specific mortality, like cardiovascular disease-related mortality.
That's right.
I mean, that was
Yeah, yeah.
pretty
I
interesting
mean,
to
just to set the stage. So we have some epigenetic clocks whose purpose is really to measure chronologic age, period. But then we have other epigenetic clocks that are really defined to be lifespan predictors, or they are meant to predict time to death or time to major onset of a disease, or what people call health span. How long are you healthy? And as you mentioned, we have actually two biomarkers. One is called DNA methylation PhenoAge, but also another one that was named after the Grim Reaper, so called DNA methylation GrimAge. Now these biomarkers were developed really for that purpose of predicting health span and lifespan, as opposed to measuring aging. And the reason why we have these different tools is that we, of course, plan to use them in human clinical trials of anti-aging interventions, you know, and in that context, you want to see whether an intervention actually resets an epigenetic clock, and that resetting then has a benefit in terms of delaying risk for various diseases. And you mentioned heart disease. GrimAge is a pretty good predictor of time to coronary heart disease. Surprisingly, these clocks even predict time to cancer. And that is surprising because it's a measurement based on blood, you know. And so you could ask, why would a measurement in blood be predictive of the onset of various types of cancers in other solid tissues, you know? But
you can predict it before other clinical diagnostics in some cases?
Yeah, I mean, let me start out by saying I'm not sure whether this biomarker is clinically useful, okay? Because I'm very scared people think they can now measure their blood and I will predict you will get cancer in 10 years. It's not at that level. However, if you have, for example, a study of 1,000 women and you measure their, somebody collected their blood in the 1990s, you know, and so for each woman you have follow-up information, whether she developed breast cancer or when she developed breast cancer. And if you then analyze the data, you will find that biomarkers such as GrimAge and other biomarkers actually do predict onset to cancer in a statistical fashion. The p-value would be quite significant, you know. But as I said, I wouldn't claim that this is right now ready for prime time in a clinical setting for finding high-risk individuals, you know, because the effect sizes are too small. You know, when I predict, for example, that you will develop heart disease in 15 years, you know, there would be a big error bar associated with it, plus minus 6 years or so. You know, so for the individual, it might not be useful.
That's a significant amount of time for a person.
Yes,
6
exactly.
years,
Yeah. Mm-hmm.
What about disease states? You mentioned cancer.
Yes.
People with cancer or Alzheimer's disease or Parkinson's disease, what does, have you measured the epigenetic age of these individuals, and does it look like it's accelerated aging?
Yes. So we looked at blood samples from Parkinson's cases and controls, and there's no question there's an age acceleration effect in blood. It's minor, it's 1 or 2 years, you know, but it is there. Alzheimer's disease, we looked at prefrontal cortex samples from the religious order study, you know, and again we found age acceleration in the prefrontal cortex. When it comes to blood samples from Alzheimer's disease, I think there might be a signal, but if there is a signal, it's very weak, you know. So, what other disease did you mention?
Cancer.
Cancer.
I'm wondering, cancer is a beast. I mean, there's so
Yes.
many different types and...
Yeah, cancer is complicated. So the exciting insight is that yes, blood methylation data indicate that blood samples collected before the person developed cancer show a slight epigenetic age acceleration. So that supports the view that faster epigenetic aging is predictive of future onset of cancer. And that finding has been validated by many groups. Problem is this association is weak. You need really a couple of thousand people to observe it.
Yeah.
But the question is, what about if we, for example, measured
tumor tissue?
Tumor tissue, the signal is huge. So if I, for example, when I analyzed malignant breast tissue samples from women with so-called luminal breast cancer, the epigenetic age acceleration is off the chart. So their breast tissue is much older than expected. But it's complicated because...
Have you compared it to their blood? Like, is tumor tissue the same?
No, it would be different. I would say that, let's say in breast tissue we find 10, 15 years age acceleration in malignant tissue, but in blood, that effect would be much smaller, if at all. Yeah.
That's it's very interesting, because you mentioned the Alzheimer's disease blood also
very weakening
signal. But if you if you measured actual brain tissue, which is where this it's a neurological disorder, you find a signal.
Yes.
The interesting thing is, you said Parkinson's disease. You do measure the signal in the blood. And
yes, that's true.
It'd be really interesting to know if the immune system is playing a role in Parkinson's disease.
Yeah, I can tell you. I can tell you the following. So we did this study of Parkinson's people. Why? Because I was interested in epigenetic aging. However, my software also produces estimates of blood cell counts. And so it turned out that the blood cell counts, in particular neutrophils, were really highly elevated in Parkinson's disease. Huge effect, And so in certain ways, this was completely surprising to me. But this finding has now been validated over and over. So yes, PD cases have highly elevated neutrophil counts.
That's very interesting.
Yes. So yes, immune system plays a role. We don't know the causal direction,
Right.
Is it first an immune dysregulation that gives rise to PD, or is it the other way around?
Yeah. There's been some interesting links to gut, the origination of the gut and the Parkinson's, and of course the immune system's involved in the gut and all that. So the reason I asked about the cancer tissue, though, was because another biomarker of aging, telomere length, the longer the telomeres are, it's associated with, basically better biological aging in a way, because your telomeres get shorter with age.
Right.
So it's assumed that longer telomeres means younger, right?
Yes.
But some cancers find a way to reactivate enzymes involved in, building telomeres, telomerase, for example.
Yes.
And so some cancers have longer telomeres.
Absolutely.
And so if you just were looking at that one biomarker, you'd look at that tissue sample and think, oh, this is,
Yeah, let me make a few comments about telomere length. So, as you said, by now we know that there is a U-shape behavior. You don't want telomeres that are too short, and you don't want to have telomeres that are too long. And so that's the first statement. The second statement is telomere length per se is actually not a good biomarker for predicting onset of various diseases. Most diseases don't have a strong relationship with telomere length. In particular, when it comes to predicting lifespan, telomere length is actually a shockingly weak predictor of lifespan. For many years, people wrote articles where they claimed there is no relationship to lifespan. By now, the field has moved on to say, well, if you have very large data, you do see a relationship to lifespan. But if we compared telomere length versus an epigenetic clock such as GrimAge when it comes to predicting lifespan, time to cancer, time to coronary heart disease, there would be no comparison, So in this sense, telomere length plays a very important role in certain disorders, but it's just not a broad biomarker for aging.
Right. How does the epigenetic clock, whether we're talking about the PhenoAge or DNA GrimAge, relate to other biomarkers of aging? So does it usually correlate, like if you have, so there's immunosenescence, which is associated with aging, DNA damage, inflammation, there's telomere length. Does it correlate typically, like in the same direction?
Yeah, it would. So talking about GrimAge or PhenoAge, they would correlate in a consistent fashion. So they would have a weak correlation with telomere length. To give you a number, correlation 0.1. So it's actually a weak correlation, but yes, if you have 1,000 people, you pick it up. In general, telomere biology is really a different hallmark of aging compared to epigenetic changes. So they measure different aspects of aging, but yeah, there's consistency.
Telomere biology has been. I interviewed Dr. Alyssa Epel on the podcast. She works closely with Dr. Elizabeth Blackburn.
Yes.
She's at UCSF, and she has published some studies showing that like stress plays a big role It plays a pretty good role in telomere biology. So you can find that like different types of stress can actually affect telomere length.
Yes.
So lifestyle factors that affect the epigenetic clocks. So for example, diet, exercise, smoking, or even
education.
Education, exactly.
Yeah.
So how do those lifestyle factors in general affect epigenetic aging?
Yeah, everything your grandmother ever told you about living a healthy lifestyle is kind of corroborated by our epigenetic clocks. So for example, people who eat vegetables, people who exercise, also actually educational level, to some extent even alcohol consumption, show a beneficial effect. Now, the problem is these effects are weak, again, you need a couple of thousand people, then you pick it up. In terms of statistical significance, there's no debate. These are clearly these associations are there. But for the individual, the question is, what if I follow the perfect lifestyle? Do I make a big dent on epigenetic aging? And the answer is, unfortunately, not really. And I mean, I'm as much of a health nut as many other people in Southern California. So I'm trying to have a healthy lifestyle, but so yes, you want to avoid diabetes and all of that, and certainly you don't want to smoke, But the truth is, a lifestyle intervention will never have a profound impact on aging at a population level, Because what I would like to do is I would like to increase health span by 10, 15 years, And in my opinion, lifestyle interventions won't get us there, In healthy people, okay? So let's say you have a friend who smokes and is obese, then yes, tell your friend to adopt a healthier lifestyle, because for this person it will have a huge effect. But let's say you take somebody like me who is reasonably slender, doesn't smoke, and now you tell me, what about if you become a vegetarian, or what if you double the amount of exercise you do? Will you have a strong effect on my lifespan? And the answer is no, not really.
According to the epigenetic.
According
clocks.
to the epigenetic clocks, yeah.
So the physical activity was only like a week.
Yeah, physical activity. Yeah, is exactly. Unfortunately, weak. So I want to say correlation 0.08 for people who know what that means. That's a very weak correlation. In blood, though.
Right.
So, because the question is, maybe if we studied heart tissue
or
muscle, maybe that we would find a much more pronounced effect, But
right.
At least in blood, we didn't see it.
Well, that sort of brings the question about tissue types too, as well. I mean,
Yeah. Example is the effect of obesity on epigenetic aging. Turns out, obese people age faster in blood. However, the strongest effect can be found in liver tissue. So obesity greatly accelerates the epigenetic age of liver tissue, you know. And so I think a lot of stress factors really have an organ-specific effect. Conversely, anti-aging interventions also have an organ-specific effect. So, for example, when we evaluated the effect of post-menopausal hormone therapy in women, we found no beneficial effect in blood. However, interestingly, the buccal epithelial cells, so the cells inside of your mouth, they actually revealed that women who took hormone therapy were aging more slowly in these cells, you know.
Oh, interesting.
Yes.
Many cell types are also epithelial cells. I mean, many of your organs have epithelial cells.
Yes.
Blood cells are a little different. I mean,
yeah.
So
and no, the finding made sense because blood cells don't have as many estrogen receptors as buccal epithelial cells.
Oh, really?
So yes, obviously, if you have a hormone intervention, you want to study cells that are susceptible to it, you know. But yeah.
Right. So that really does highlight the importance of measuring different tissue types to see, because I mean, sometimes it's easy to kind of think, well, whatever fundamental mechanism is regulating this aging process would do it in all the tissues, but it doesn't necessarily mean it will be the same either, right?
Exactly.
So it's tricky.
I mean, it would be fantastic if a blood measurement is really a surrogate for all other tissues and organs, you know. And but it seems to be not the case, at least for stress factors, as I mentioned, environmental but also even genetic factors, you know. So some people inherit a genetic variant that accelerates their epigenetic age in blood, but not really in brain tissue, you know. So even there it's complicated, yeah.
So there's genes that are regulating the epigenetics, instead of vice versa. So I kind of lost track of, I was going to ask you something. Geez, it was important too. But, I know what it was. I had read a study, I think it was one of your really good reviews that you published, where you talked about bone marrow transplants.
Yes.
Because the question is, you know, there's lots of these studies coming out with parabiosis where you can transplant young blood from animals into animals that are older and sort of have this rejuvenation effect.
Yes.
So the question is, if you take cells from a younger recipient and put it into an older bone, if you take cells from a younger donor and put it into a older recipient, so you're basically taking young cells and putting them into an older person.
Yes.
So the young cells will have a younger epigenetic age, obviously, than what was already there.
Yes.
The environment around the cells, the niche, so to speak.
Yes.
Does that play a role in? Do those blood cells have more of an accelerated aging?
Not really. Yeah. So just to be clear, I want to distinguish parabiosis from these hematopoietic stem cell transplantations, I can comment on both. But let me start out with the hematopoietic stem cell transplantation. Some people have a very severe form of leukemia, and therefore their bone marrow stem cells have to be removed. And the procedure is dangerous. I'll start out with that. So that's why I say only severe forms of leukemia patients get that treatment. But yes, let's say you take a 50-year-old and you give this person a bone marrow transplant from a 20-year-old. And so after the transplant, the blood in the recipient reconstitutes itself. The person has now new blood. And the question is, what's the age of the blood? Is that blood, does the blood have the age of the 20-year-old donor or the age of the 50-year-old recipient? And you can make a case for both scenarios. But there are now several scientific papers that really give an unequivocal answer, and which is the reconstituted blood in the recipient has the age of the donor, you know. And that effect persists for decades, you know. So if you take again the 50-year-old, got a bone marrow transplant from a 20-year-old, you follow this 50-year-old 30 years, now he's 80. Question is, how old is his blood? Well, the age would be now 50 because 30 years have passed and you add that to the age of the donor, you know. And one would think that the stem cell niche in the bone marrow, you know, so could possibly affect the aging rate, you know, but it just isn't the case, you know, and so that's on some level a very exciting finding, because it kind of hints to an idea that you could possibly rejuvenate people through transplantation, you know. The reason why this is not yet a viable strategy is because people who get a transplant often get so-called graft versus host disease. So there are all sorts of complications, you know. It's a dangerous procedure, but in theory, you know, it could work, you know. And talking about parabiosis, where people have connected two mice, you know, where one mouse is much older than the other one, or much younger than a mouse. We just recently analyzed parabiosis mice, you know, it's unpublished, and we found two results. One corroborates things and one refutes it, okay? But just to explain it, so we looked at cortex and also subventricular zone, deep white matter in the brain, and we found that mice that were a young mouse that was connected to an old mouse actually aged faster according to an epigenetic clock in mice. So that part confirmed these parabiosis experiments. So in other words, you can age a young mouse. But that's not what people are interested in. They're interested in the opposite. You take an old mouse and you connect it to a young mouse, and then you study the brain of the old mouse, and you want to see that the brain is rejuvenated, you know. And for that scenario, actually, our results were disappointing. We didn't see a rejuvenation effect, you know. And so now we're trying to get additional data, because our first study was underpowered, but one of these months we will have a definitive answer. You know.
Yeah. For, you know, some of these animal studies are really good for trying to understand mechanism, and
yes,
all of this data suggests, you know, you've got a clock that can predict chronological age. You've got a clock that can look at your biological age and also predict time to death, lifespan.
Yes.
The GrimAge.
Yes.
And I mean, something clearly is changing these methylation patterns.
Yes.
So the question is, what is that? Is there a chronic signal that's doing it, or is it just completely under genetic control? Like, and
yeah,
maybe they're related, right?
Yes.
So what are your thoughts? What are your thoughts on the aging process, and even...
Yeah, I mean, well, when it comes to these epigenetic clocks, this is the #1 weakness of these clocks, that we don't completely understand the molecular mechanism. And coming back to telomere length, that's a great advantage of telomere biology. We really understand very well what regulates telomere length, you know. But yeah, with the epigenetic clocks, this is a very active area of research. Top biologists and labs are working on that very question, you know. And there are many theories. Some people think stem cell biology plays an important role, and that's probably true for many tissues, you know. In certain ways it could measure aspects of stem cells, for example, how often a stem cell divided. And the problem with that interpretation is that the epigenetic clocks work beautifully in neurons, you know, which really don't rejuvenate over the lifespan, you know. And yes, another group thinks that epigenetic clocks might relate to circadian rhythm, so there have been some theories. Some people now, I believe that also processes that play a role in development must be playing a role here, and the reason is because my original pan-tissue epigenetic clocks works actually beautiful in prenatal brain samples. It works beautifully in various in vitro studies of so-called three-dimensional brains, you know, or in retina samples. So really, it captures aging of gestational age during development, you know, and during development, there's really no noise, you know. This is a highly coordinated process. And so yes, these processes also must play a role, you know. Yeah.
Yeah, certainly. One thing I had thought about, and I want to get back to development because of the stem cell thing, but one thing that came to my mind with this chronic signal is one particular gene that is methylated during early age, but then becomes demethylated as a person ages, is this P16 INK4A gene.
Yes.
And it plays a role in cell cycle progression, meaning it it basically stops the cell from going on to the next cycle.
Yes.
So obviously you don't want it to be active during development or early age because you want your cells are growing and but it also, so when it becomes active, it stops stem cells, like hematopoietic stem cells, they aren't growing.
Yes.
So there's like this kind of role in aging where it's sort of basically stopping the stem cell from growing is going to have a negative impact on aging, although it can be positive for cancer because, well, positive for the person that has cancer because it can stop a cancer cell from growing, right?
Yes, yes.
So the question is, there's a group of demethylases that can take off the methyl group, that become active and take it off of this gene, and they're activated by inflammation. So what I'm wondering is anyone looking at, obviously these methyl groups are changing, and so the enzymes that are pulling off methyl groups and the demethylases, the enzymes that are putting them on their methyltransferases, they must be doing something.
Yeah, and people are looking at it.
What's changing those enzymes? Like, is there a signal there, or is it a gene? Is it genetic control, or what is it?
Yes.
That can change.
Yeah, you make very good points. So, if you want to understand the epigenetic clock, clearly you start with so-called DNA methyltransferases, or these TET enzymes. Why? Because they, on the one hand, add methyl groups or remove methyl groups. So that's the low-hanging fruit. And just recently, we and others, several groups, have actually found, no doubt, these, when you interfere with these enzymes, you affect epigenetic age. They're very exciting findings where people studied certain developmental disorders where mutations deactivated a DNA methyltransferase, or a mutation rendered it overactive, And sure enough, all of these mutations in humans, affect epigenetic age. And so at that level, we know it has an effect, as expected. And the effect is pronounced. It could add 5 or 10 years to a person, or the opposite,
And does that correlate with the lifespan of what?
See, we don't know exactly. That's the question. So some of these children have a developmental disorder, and there's various syndromes, one is so-called Sotos syndrome, and we see strong deviations in blood in both directions, +5 years, -5 years. So at that level, it's all confirmed. Plus, we right now have we do mouse crosses, where we knock out these DNA methyltransferases, and just to very carefully study it in a very controlled setting. But I can already tell you, you'll find strong effects. Now, but the question you really ask is, well, on this an upstream of those, what regulates the clockwork? What tells the DNA methyltransferase go to this location, and deposit and work your magic,
Right.
And that's where we don't have an understanding yet.
And that's why I mentioned the Jumanji demethylases, because that is just one group of demethylases that's particularly plays a role in taking off the methyl groups for the INK4A locus. And I remember trying to figure out, inflammation plays a role in aging, and so I had seen studies showing, sure enough, TNF alpha, these things play a role in activating those demethylases.
Yes.
And so it's like, well, is that something that's just over time, chronic, that there's a threshold, or what, I don't know.
Absolutely.
I've never seen the experiment done with that particular
classic
enzyme.
I've seen results from other groups, you know, that look at that issue, chronic inflammation, or even looking at these what are these? Transposons, you know. So there's now some very exciting results that show that some transposons become active in older tissues, you know. And so I've seen some preliminary data where people said this was associated with increased epigenetic aging. And also our finding that HIV is very much associated with accelerated epigenetic aging also points again to this idea of a viral component, you know. So clearly there must be a connection, you know, and it will be very interesting to tease it out, you know. Right. I don't have a good sense how strong that effect is, you know. Does, for example, chronic inflammation, or do these enzymes that you mentioned, you know, how strong is their effect if we perturb it? Do they explain 30% of the variability or just 10%, you know? So that's in certain ways the question. Yeah.
Have you measured the epigenetic age in stem cells versus already differentiated cells? Is there a different pattern or a different...
Yeah, there's a different pattern. So if you take, for example, a stem cell, an iPS cell, and then differentiate it into a more mature cell, for example, a more mature neuron, you will find that the epigenetic age increases. The issue is it doesn't increase by a lot, you know. So a more differentiated cell, a more mature cell, may be maybe 1 or 2 years older than the stem cell. You know? The question is how do we age a cell by 30 years? You know, because when you study neurons in a dish, you want to perhaps understand neurodegeneration, and so you want to study very old neurons, you know. And so when you deal with neurons derived from stem cells, we don't have a good way to age them. But nowadays people pursue this idea called transdifferentiation. So you take, for example, a skin cell and you add certain factors, maybe microRNAs or what have you, and then turn the skin cell into a neuron. And this transdifferentiation protocol actually preserves the epigenetic age, you know. And so we have shown in collaboration with several groups that yes, the resulting neuron has the epigenetic age of the skin cell.
Oh, that's interesting.
Yes.
So what about when you take a skin cell, and you mentioned induced pluripotent stem cells, and you basically can add, you know, like 4 transcription factors, I think even less now, but the original from Shinya Yamanaka, 4 transcription factors, and make this into like a pluripotent stem cell that can become any type of cell in the body. The epigenome, what happens to the epigenome?
You completely reset it. I mean,
to a completely young.
Completely young.
So that's like the ultimate.
Yeah, it's the
reversing aging, right? I mean,
it's true, because in the past people were wondering, you know, are there interventions that actually reset the epigenetic clock? And so, and the number one proof of principle study is really the administration of these Yamanaka factors, because it completely resets the age, actually to a prenatal stage, you know.
And that's completely genetic, right? I mean,
these are
manipulating genes.
Yeah, I'm not sure. Yeah, I mean, or it's really so also messenger RNA can do it, you know.
Sure. I guess what I mean is it's something that's kind of under a genetic control.
Yes.
Like ultimately.
I mean, right now there's an idea in the aging field to rejuvenate people by leveraging this fundamental insight that it's called reprogramming. You can take an old cell, you administer certain factors, as you mentioned, 4 factors or 3 factors or other variants, you know, and by administering this cocktail of factors for just a few days, not too long, because if you administer it for too long, you greatly increase the risk of cancer, you know, and because the cells lose their identity. The skin cell forgets that it's a skin cell. It thinks it's a stem cell, you know. But if you do it briefly for, let's say, 5 days, you get the benefit of rejuvenation. You may have rejuvenated the cell by 5 years or 10 years, but it still remembers its identity, And
so does the epigenome reset a little bit? Like it only...
Yes, yes. So there are a couple of groups that are working on it and have already shown that effect, Yes. So I analyzed fibroblasts and endothelial cells from such an intervention. It's called sometimes interrupted reprogramming or transient reprogramming, and sure enough, that idea worked. It reset the age, but the cells still remembered their identity and therefore arguably will not become malignant.
Right.
Yeah.
Yeah, that is super interesting.
I have to say that's true. And the epigenetic clock is the ideal biomarker for that kind of a study, in my opinion, because as I said, it very much tracks, the state of the cell, but we will see, hopefully in 5 or 10 years, it turns out that this strategy turns into a viable anti-aging intervention that can be used in the clinic,
Right.
But at this point, these are all proof of concept studies that have been done in the dish or in mouse studies,
Do you personally think that the epigenetic clock plays a causal role in aging? Do you think?
Yeah, let me answer it in 2 ways. It clearly relates to a process that plays a causal role, and so to use a metaphor, is it the face of the clock, or is it the clockwork of aging, And no doubt the epigenetic clock must relate to at least one causal process because it predicts lifespan, If it didn't relate to a causal process, it wouldn't be able to predict how long you live. Now, but the real question is perhaps, what if you changed the methylome? If you had an intervention, you changed DNA methyltransferases, you kind of, I want to call it the superficial way of perturbing the clock. Would that have a benefit? And personally, I don't have an answer to it yet.
Yeah. Have you ever looked at how? So you mentioned obesity and how obesity is associated with an accelerated epigenetic age.
Yes.
Have you ever come across data with respect to the opposite of that, like fasting or even caloric restriction, if that slows the epigenetic aging?
Yeah, I mean, in mice, the answer is clear-cut. Definitely caloric restriction slows the epigenetic clock in mice. And we know that because several groups have looked at it, including my group. All of us arrive at the same answer. Conversely, by the way, high-fat diet, accelerates the epigenetic age of mice, so that's all clear-cut, The question, though, is really, what about humans,
Right.
And I'm not aware of a study where people really looked at caloric restriction versus epigenetic aging. And but let me share some thoughts. When it comes to any intervention, including dietary intervention, that prevents anything related to metabolic syndrome or diabetes, that will be detectable, So in other words, if you have an intervention that takes a very obese person to a lean person, in my opinion, GrimAge will pick that up. It has to, However, when you ask the question, what about if you take a relatively lean person, healthy body mass index 23, and and this person is so motivated to live 10 years longer, and they pursue now a lifestyle where their BMI is, let's say, 17, almost at the point of being unhealthy. And so if you compare a healthy person versus a semi-starved person, would that have a benefit? And in my opinion, that may not be the case. And I'm just speculating here. But even in according to other biomarkers, I haven't seen convincing studies. Otherwise, I think my BMI would be 17 right now. So yes.
There's an acquaintance of mine who has, he runs a pretty popular aging blog. His name is Josh Middledorf. He's reached out to me, I don't know, a few months ago, because he's trying to organize a study where people that are basically already practicing some type of fasting, whether it's an intermittent fasting, they're doing, you know, a 16 or 24-hour fast, or sometimes maybe they do prolonged fasting, which in some cases can be, you know, longer than 48 hours, 48
Yes.
hours or more.
Yes.
People are doing this stuff, you know, it's just it's happening.
I'm trying to do this intermittent fasting, but I don't have much self-discipline. But yeah, we will now analyze mice, you know, from the lab from Joe Takahashi, who did various interventions, and probably in a couple of weeks we will have some answers, you know, whether these strategies have a strong effect in various tissues in mice, you know.
Oh, cool.
But yeah, when it comes to human data, that's really
Right.
the prize, you know.
It is. And as you
mm-hmm.
mentioned, you know, you have someone that's starting off with already what you would think is healthy, someone who's not obese, someone who exercises, doesn't smoke, someone who, you know, is, you know, eating a relatively good diet. You know, if you take that person,
Yes.
who, you know, is already pretty healthy,
Yes.
and then do an intervention. So is that intervention is going to be whether it's like a intermittent fasting. So I mentioned Josh Middledorf because he wants to track, you know, there's a company that does based off of your clock. I don't know which clock, the Horvath original clock or the PhenoAge, but they're called My DNAmage. I just ordered
that's
their.
right. Oh,
I just
really?
ordered it,
I
so I'm
see.
going to get. it just arrived. I'm going to get my blood tested.
It's the original clock.
It's
of
the
mine.
original.
Yeah,
Okay, so
except
that's-
they have modified it a little bit, you know. So I think, yeah.
So that's probably better at
Yeah.
predicting chronological age rather than biological age.
Well, I think nobody knows, you know,
Nobody
because
knows.
these
Right, because-
these biomarkers should really be
Right.
evaluated in a prospective study. but
Yeah.
mhm.
Because so that's the question. It's like, well, if you have these types of interventions on already healthy people, will they make any difference? At least
Yes.
according to
Yes.
epigenetic aging, right? Doesn't
Yes.
mean that.
Yes.
I mentioned before we started filming, I mentioned to you a pretty recent randomized controlled clinical trial with vitamin D supplementation in a population that started off unhealthy. So these were obese African Americans, which were very low in vitamin D. African Americans tend to be the lowest in vitamin D because they have a natural sunscreen. And so they were given 4,000 IUs of vitamin D a day, and after, I can't remember how long the trial was, a certain period of time, their epigenetic clocks were measured at baseline, and after, and their epigenetic clocks, the Horvath clock was used. It basically was reduced by 1.8 years or something like that. And you mentioned the significance is a little, the sample size is small, and so it's kind of like,
I mean,
well,
it's an
it's
exciting finding. I think it was based on 51 people,
Yeah.
and it's a nice finding. It would be brilliant if a simple intervention as taking a vitamin D supplement actually affected the epigenetic age. I think I agree with the authors. We just need larger studies to validate it, you know. But in general, you know, as you know, clinical trials are very expensive, you know, and that's a real bottleneck in all sorts of anti-aging interventions. I really wish, on the one hand, private industry would find merit in investing in clinical trials. Obviously also the government plays a role, but what we really need is really dozens, if not hundreds, of clinical trials. Why? Just to have a chance for serendipity. Maybe it is as simple as a vitamin D supplement, but maybe you need something much more radical, like, for example, a modification of the Yamanaka cocktail, you know.
Right.
Or it could be plasma transfusions or so. You know, we as a field, we need to experiment, you know, with what kind of interventions work. Could be hormones, by the way.
Right.
You know, it could be a hormone intervention.
Well, you mentioned
so on,
obviously.
yeah.
Do you see? I'm going to change topics here, but I don't want to, because you know the intervention trials are important. You know, as you mentioned, it may have some effect. It may not have a big effect, but you know, even if a combination of factors like getting your fish intake, which you've shown to also,
Yes.
seems to be related to.
Yeah, interestingly, according to GrimAge, we did find that people who used omega-3 supplements or fish oil, they were actually aging more slowly. And we thought this was a nice little insight.
Oh, that's interesting, because
yes.
I take fish oil supplements.
Me too.
Yeah.
Because what happened is, I want to say 6 months ago, there was a publication, really very large-scale clinical trials looking at fish oil supplementation, and they did not observe any benefits. And I said, oh my God, I wasted all these years eating fish oil. But then we analyzed really an observational study, and that's our problem. Our study was an epidemiological study. I want to say the Women's Health Initiative. And there we did see this association that women who took fish oil supplements were aging more slowly, according to GrimAge.
Wow.
Yeah.
And I'll just add a note to that study you mentioned, the big Vital D study, which was a vitamin D and omega-3, a huge study.
Yes.
There was no effect on the primary outcome, which was
Yes.
looking at all combined cardiovascular related events.
Right.
However, when you specifically looked at heart attack versus stroke, there was a strong effect. So
I see.
it's always this, the secondary outcome, like however
Yes.
you're designing the trial,
Yes.
there's a primary outcome. If that primary outcome is negative, then it's like, well,
The problem with the primary outcome is that it's far removed from the processes that are being targeted by the molecules,
I agree.
And that is really the benefit of having what I call a surrogate marker, something like GrimAge is arguably much closer to the mechanisms, is perhaps less susceptible to confounding factors, and so maybe that's why these surrogate markers pick it up,
Right.
Mm-hmm.
Yeah, I mean, obviously the methylation patterns, these are molecular changes that happen before someone has a heart attack, before someone has high blood pressure. I mean, these are accumulated changes at the molecular level that are occurring.
Yes. And also, when you think about heart disease, it probably relates to many confounders such as psychosocial stress. You go through a divorce, you get fired,
That's true,
various substance abuse, a lot of processes that are in certain ways stress factors, but they are not part of what I would call innate aging processes, whereas these biomarkers, such as an epigenetic clock, is hopefully much closer to an innate aging process, And when we want to cure aging, we want to cure innate aging. We don't want to, it's not my goal to reduce people's stress levels, For that, they should do yoga or any other intervention,
Right. Sleep, I mean, obviously sleep is another thing associated with heart attacks and
Exactly.
I don't know if sleep is even usually one of the confounding factors that's adjusted for, right? I don't even think that's something I usually
we
see.
looked at people, women who have sleep disturbances in the Women's Health Initiative, and sure enough, their epigenetic age of blood was slightly accelerated.
That makes sense, yeah. I talked to a sleep expert not long ago, Dr. Matthew Walker. He runs the Human Sleep Center at UC Berkeley, and he just talked about all the, basically various diseases and, all-cause mortality and how everything just goes up when sleep quality goes down, and so I certainly feel like...
I always hate these studies because I don't sleep well. I like the studies where they study the so-called super sleepers, who sleep only 5 hours a night and still are perfectly healthy. So I like more the optimistic spin on things. But also, just to tell you, so the effect of sleep quality, versus epigenetic aging in blood, these associations were statistically very significant, but again, they were weak, and that makes me hopeful.
Yeah, compared to the semi-supercentenarians, which had a epigenetic age of 8.6 years younger than their chronological. That's pretty robust,
that's
right? Would you that would be a robust or more strong, I guess.
Yes, I want to say it was maybe not 8 years. I want to say it was 5 years. So it depends on what you compare.
Not the offspring, but the actual person.
That's right. Yeah. So you're right on. So if you analyze the blood from a centenarian or supercentenarian, it's true our age estimates are really way below their chronologic ages, could be 15 years younger, you know. So there's a real leveling off effect, you know.
Yeah. What about studying how other there's other biological processes that, at least in animals, when you perturb them, are known to regulate aging. For example, you can mutate certain mitochondrial factors and have accelerated aging, or you know, cellular senescence. You can also have a certain in mouse where you can accelerate aging, or the opposite, where you lower growth hormone levels and they'll you know, they live longer. Have you or anyone looked at the epigenetic aging, how that relates to these other?
To some extent, yeah. What you mentioned, these growth hormone knockout mice that are known to live longer. Sure enough, according to the epigenetic clocks in mice, they really age more slowly. So it was a beautiful validation, you know, of the fact that epigenetic clocks measure biologic age, because growth hormone receptor knockout mice, that's really a gold standard anti-aging intervention, and you want that a clock picks that up. And when it comes to other strategies, senescence, right now it's a hot topic, these so-called senolytics that remove senescent cells, you know. And we are about to analyze data collected by James Clement, who looked at, who did clinical trials of these senolytic strategies. And hopefully we'll have an answer in a couple of weeks, you know, but I just don't know whether removing senescent cells has an effect on epigenetic age. You know. In general, the relationship between senescence and epigenetic age is complicated, because when it comes to inducing senescence, there's several ways of inducing senescence. One is simply what is known as replicative senescence. You passage cells, you split them and let them grow and grow and grow. And that form of senescence is somewhat related to epigenetic clocks, you know. Then there are other forms, so-called radiation-induced senescence. You radiate a cell, and that form of senescence doesn't seem to accelerate the epigenetic clocks, you know. So it's complicated. And conversely, there are ways of immortalizing cells by overexpressing this component of the telomerase, the TERT. Now that, so immortalizing a cell actually doesn't stop epigenetic aging, you know. You can have an immortalized cells that you can passage for decades, but the epigenetic age keeps going up in the cells. Yes. So do you see, epigenetic clocks are not simply markers of cellular senescence. They really pick up a different aspect of biology.
The radiation and DNA damage is kind of surprising, because I think in one of your papers looking at, and this was another question I wanted to ask, was where are these methylation patterns occurring in the genome? Are there genes that are really particularly known to be involved in the aging process, you know, like, or in, you know, basically in just being health in general?
Yes.
Like, are these, like, you know, the metabolism genes or DNA repair and things like that?
Yeah, I wouldn't make that claim. So let me start out by saying, so my original clock used 353 loci. When we look at GrimAge from Akelu, my scientist in my lab, it uses over 1,000 locations in the genome. And now, one question is, what if we removed these locations from our data and just built a new clock? Would we still get another good clock? And the answer is yes. You know, I could have built alternative clocks using other locations in the genome. You know, on that level, these locations are not unique. You know, and when you look at the genome, we have, in principle, 28 million locations in the genome. Are cytosines, you know, and I want to say a quarter of them change with age. Some of them gain methylation, some of them lose methylation. So these methylation changes are almost global, you know. And in that sense, epigenetic clocks look at perfect representatives of the entire, what is known methylome. They represent everything that's going on. But you can see that maybe looking at only 300 locations is not ideal, you know. Having said this, we certainly did look at it and say, are these locations enriched, you know, with certain pathways? And no doubt they are, you know. So sites that gain methylation with aging are known to be located in so-called polycomb group protein target sites. So certain proteins that play a very important role in maintaining stem cells, you know, or conversely, sites that play a role in cell differentiation and development. So these sites tend to gain methylation with aging, you know. The sites that lose methylation also are enriched with certain themes. For example, often they're in so-called enhancer regions, you know. So the field of epigenetics has very much characterized the genome, which parts change with aging. There's wonderful review articles on it.
Yeah.
Mm-hmm.
Yeah. And then I just got on the stem cell thing. That's just so interesting that a lot of those are regulating stem cell function because it's just
coming back to the mechanism of the clock. That's really a profound insight, you know, that when you look at the data, you keep seeing themes related to development, tissue differentiation, organ development. And it is a profound insight because if you had asked an aging researcher 5 years ago whether developmental processes matter in aging, they would have said no, you know. Many people think of aging as noise or wear and tear, you know. But these epigenetic clocks have really linked development to tissue dysfunction in a direct manner. An epigenetic clock is a continuous readout that links prenatal tissues directly to very old samples.
It's really
you know?
blowing my mind. It's actually, that is, I
combines
never would have
two
thought.
fields,
It does,
you know? Because when many people obviously study development, but these are not aging researchers,
no, of course,
but these clocks really point to commonalities, you know, of these fields. Yeah.
Wow, it's just, it's amazing. It's very interesting. I just want to thank you so much for doing all the work that you're doing, and will continue to follow, you know, your work.
Thank you for your interest.
People that want to learn more about your research, probably the best place is Wikipedia. We talked about. I mean,
I've written a review article in Nature Reviews Genetics. People have written Wikipedia pages. Then there, you mentioned the blog by Josh Mitteldorf. You know, so there are various forms of learning about them. You know, so.
Well, thank you, Steve, so much. I've really enjoyed this conversation.
Great, thanks.
Oh, hey, you're still here. I'm just sitting here mixing up next week's Found My Fitness Premium membership benefits. And they're
spicy.
That's right. Almost every week we send out something cool, like our bi-weekly Science Digest, where we summarize the top news stories in science of the week. Or our Q&As, which happen monthly. Or the Aliquot, which is our members-only podcast. We take clips from cool interviews like this, and we add a little extra information. If you want to find out more about becoming a Found My Fitness Premium member, you can click here. That way we can make more cool videos like this one. Catch you next time.